Hey everybody, so we're going to do something a little bit differently with this video. We're going to do some live action, and we're actually going to do an activity that we would have done in class. Obviously, we're all stuck at home, so I've recruited my wonderful daughters, Char and Mads, who are going to be helping us today in place of you guys. And they will be the receptors, the membrane itself, translocation channels, etc. So this whole video is about how to get proteins into the ER membrane so they can eventually be shipped off to the Golgi membrane or the cell membrane, wherever they need to go. So the first story we're going to tell is just a regular secreted protein. It's going to have no transmembrane domains at all. It's going to be made at the rough ER. It's going to fold up inside the lumen of the rough ER, and then it'll be loaded as cargo into a vesicle and secreted from the cell. And the rest of that story, vesicle formation and vesicle docking and fusion, is told in the other video for this week. So let's tell that story first. So we're going to start off with a ribosome, and that ribosome is going to be making a new protein out of a messenger RNA, like we talked about in Unit 1. And the very first thing to be made by that ribosome is the signal sequence. That's the first part of the protein that's being made. And as soon as that signal sequence is made, it's bound by the signal recognition particle, or the SRP, and it's going to carry this whole complex over to the SRP receptor. So now the SRP receptor is holding on to the beginning of the protein that was being translated. But here's the ribosome now tethered to the ER membrane, making it rough. That's why it's the rough ER. That SRP receptor is going to transfer all of that to a protein translocation channel, and we continue to translate. So this is the protein being made from the ribosome. It's being threaded directly into the ER. We hit a stop codon. Translation terminates. The ribosome floats away. And the SRP, the signal sequence is severed from the rest of the protein. And then the protein will fold up in the lumen, or the inside of the rough ER, where it can eventually be loaded as cargo into a vesicle. The signal sequence itself is eventually released by the channel, and it's going to be degraded into single amino acids and recycled. The next part of this story is going to start the same way. We have a ribosome. It's bound to a messenger RNA in the cytoplasm, and translation begins, and the first thing to be made is the signal sequence. It's the first thing to come out of the ribosome. And the SRP, the signal recognition particle, is going to bind to that signal sequence, bring it over to the SRP receptor. The SRP receptor is going to transfer that over to the translocation channel, and translation will continue along the way until a stop transfer sequence is encountered. And that's going to kick that stop transfer sequence out of the translocation channel, and it's going to embed itself in the hydrophobic portion of the membrane itself. So here Mads is the membrane. That is stuck now in the membrane, so the ribosome has no choice but to continue translation in this direction, leaving an external domain. So this will fold up into a domain on the outside of the ER. And once we sever the signal sequence from the rest of the protein, we'll have another domain of this protein inside the ER. So we've got two domains in this single transdomain protein. The third story we're going to tell is for a multi-transmembrane protein. Ribosome binds to a messenger RNA in the cytosol. First thing to be made is the signal sequence, just as before. It's bound to the SRP, brought to the SRP receptor. It's getting pretty boring. That now transfers to the translocation channel. Translation continues. We're threading into the ER. We hit a stop transfer sequence, and that's ejected from that membrane, from that translocation channel, and it's now embedded in the membrane. I'm going to sever this now, just so Char has a chance to do her second roll of this little act. But translation continues outside the ER. We make this domain here, and then ultimately we're going to create or translate a start transfer sequence, which is going to be embedded into a new translocation channel. We'll continue translation until a stop transfer sequence is made, which will be ejected out of the channel into the membrane, and we continue making new protein outside the ER until ultimately a start transfer sequence is made yet again, which goes into a new channel, and we continue making protein until we hit a stop codon. And we wind up with a 1, 2, 3, 4 transmembrane domain protein, and we have multiple domains inside the lumen of the ER, like this one here, and this one here, and this one here. And we also have domains outside of the ER. When this protein gets transferred to the cell membrane, this will be exterior and interior to the cell. So I hope that it's a little bit clearer how proteins become embedded into the ER membrane, both as single transmembrane proteins, as single proteins for secretion that will ultimately be loaded into a vesicle as cargo, and as multiple transmembrane protein domains as well.